Hello everyone, in this video I will briefly introduce our research work LinearTurboFold. As we know that functional RNA have conserved structures, and their structures are even more conserved than their sequences. Here is an example. There are two tRNAs. Their conserved structures looks like the clover leaf, but their sequence identity is pretty low. It's only about 0.4. If we use algorithm to fold these two sequences separately, we got a totally different structures compared to the conserved structure. And if we align these two sequences without considering any structure information, we got poor alignment due to the low sequence identity. The colors correspond to stems, but the stems are not aligned very well. To solve those issues, we need algorithm to jointly fold and align our homolog sequences. And based on the structural alignment and predict structures, we can extract the complementary mutations, which are strong signals for conserved structures. Here is the LinearTurboFold. LinearTurboFold analysis homolog sequences. There are two linear modules, linearized alignment and linearized folding. Linear alignment utilizes structure information to generate structure alignment. Linear partition incorporate the structure information from homologs and also utilize the alignment information to predict conserved structures. LinearTurboFold scale linearly with the sequence lines. SARS-CoV genomes like SARS-CoV-1 and SARS-CoV-2 belongs to coronavirus genomes, which have the longest viral genomes. Existing work are too slow to scale to coronavirus genomes, while LinearTurboFold is the first join, fold, and align algorithm to scale to full-length coronavirus genome. Also, LinearTurboFold significantly improves folding accuracy and achieves comparable alignment accuracy. As I just said that coronavirus genomes have the longest viral genomes, most existing work for modeling SARS-CoV-2 structures have to use the local folding methods with short window size and limited base pairing distance, which totally ignores the long-range interaction. But instead, LinearTurboFold enables unprecedented global structural analysis on SARS-CoV-2. Over a group of SARS-related 2 and SARS-related genomes, like SARS-CoV-1 and bad coronavirus, we predict the conserved structures. We observe that our in silico prediction is close to experimentally guided model for local structures. Furthermore, we can capture long-range interaction and predict end-to-end pairs between 5' and 3' UTRs across the whole genome, which match perfectly with a purely experimental study. All those complementary mutations extracted from the homologs indicate that those structures are conserved, and all those conserved structures predicted by LinearTurboFold are potential targets for small molecule drugs and antisense oligonucleotides. LinearTurboFold can also help to identify accessible and conserved regions. We noted that SARS-CoV-2 genomes have a pretty high sequence identity. There are only about 1% and 2% mutations. If we predict their structures separately, using algorithms, we got totally different structures. That means single sequence predictions is sensitive to the mutations. But folding with homologs like LinearTurboFold can help to predict structurally conserved accessible region. To evaluate the accessibility, we utilize a stochastic sampling algorithm, linear sampling, to sample independent structures based on the homolog where partition function dumped by LinearTurboFold. And based on the sampled structures, we calculate the accessibility for regions at least 15 nucleotides long. In addition to high accessibility, we also require the high conservation among SARS-CoV-2 and SARS-related genomes. In the end, we can get both accessible and conserved regions. And all those regions are promising targets for siRNA, antisense oligonucleotides, CRISPR-Cas13, and RT-PCR primers. Thanks for watching. If you are interested in our work, please check our manuscript and our released code and results on SARS-CoV-2. Thanks for watching.